Molecular evolution. Concepts of neutral evolution. Maximum detail paragraph format note tables CSIR-JRF level based on previous year questions full-length coaching material. The concept of neutral evolution represents a fundamental shift in our understanding of molecular change in populations, as it challenges the previously dominant view that all evolutionary changes are driven by natural selection. Introduced by Moto Kimura in 1968, the neutral theory of molecular evolution proposes that the majority of evolutionary changes at the molecular level, such as changes in DNA and protein sequences, are not caused by Darwinian selection, but by random genetic drift of selectively neutral mutations. These are mutations that do not significantly affect an organism's fitness and therefore do not influence reproductive success. In this theory, molecular evolution is described as a stochastic process. Random mutations arise in the genome, and while many are deleterious and quickly eliminated by purifying selection, a significant proportion are neutral and can become fixed in the population simply due to chance events. According to Kimura, the rate of molecular evolution is equal to the mutation rate of neutral mutations, implying that this rate is independent of population size, a key insight that diverges from the traditional selectionist view. The neutral mutation rate K is given by the formula K equals mu, where mu is the neutral mutation rate per gene per generation. This elegant relationship allows molecular clocks to be calibrated, since the accumulation of neutral mutations can be used to estimate divergence times between species. The theory explains why molecular evolution appears to occur at a relatively constant rate, even when morphological evolution is episodic or punctuated. One important concept arising from neutral evolution is the nearly neutral theory, proposed by Tomoko Ota. This refinement accounts for the fact that many mutations are not perfectly neutral but only slightly deleterious or slightly advantageous, and whether they are fixed or lost depends on the effect of population size nay. In small populations, even slightly deleterious mutations may become fixed due to drift, whereas in large populations, selection is more efficient in removing such mutations. The neutral theory has profound implications for polymorphism within populations and divergence between species. For example, synonymous substitutions, mutations that do not change the amino acid sequence, are often neutral, while non-synonymous substitutions may be subject to selection. The ratio of non-synonymous to synonymous substitutions, deci-Newton per deci-Siemens or Ca per kilosecond, is a widely used metric to detect selection at the molecular level. A ratio near 1 implies neutrality, less than 1 implies purifying selection, and greater than 1 suggests positive selection. Moreover, molecular data support the neutral theory through observations such as high levels of genetic variation within populations that do not correlate with differences in fitness, a linear relationship between molecular divergence and time. Molecular phylogenies often reveal clock-like patterns despite diverse life histories and environments. Importantly, the neutral theory does not deny the role of natural selection, especially at the phenotypic level, but it emphasizes that most genetic variation at the molecular level is not subject to strong selection. This concept helps reconcile the high mutation rates observed in molecular data with the relatively slow pace of adaptive phenotypic evolution. In modern evolutionary biology, the neutral theory serves as the null hypothesis when testing for evidence of selection. Techniques such as the McDonald-Kreitman test and Tajima's D statistic are used to detect departures from neutrality by comparing observed genetic data against expectations under a neutral model. In sum, the concept of neutral evolution revolutionized molecular biology by suggesting that drift plays a dominant role in shaping genetic variation at the molecular level, and it remains central to the fields of population genetics, phylogenetics, and molecular clock studies. Previous year CSIR net questions. According to the neutral theory of molecular evolution, the rate of molecular evolution is proportional to population size. B independent of mutation rate, C proportional to the rate of neutral mutations, C proportional to the rate of advantageous mutations. Answer: C proportional to the rate of neutral mutations. CSIR net Dec 2018. Which of the following supports the neutral theory of molecular evolution? A high Ca per kilosecond ratio. B rapid changes in phenotype. C constant molecular clock rate. D lack of polymorphism in populations. Answer: C constant molecular clock rate. CSIR net June 2017. The ratio deci-Newton per deci-Siemens in GT 1 indicates a neutral evolution. B purifying selection. C positive selection. D genetic drift. Answer: C positive selection. CSIR net Dec 2016. Let me know when you're ready for the next lecture in molecular evolution, such as molecular clocks, rate of molecular evolution, or construction of phylogenetic trees. Each will be covered with full detail as per CSIR-JRF standards. You said. Molecular divergence in molecular clocks. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on molecular evolution, molecular divergence in molecular clocks. Maximum detail paragraph format note tables CSIR-JRF level based on previous year questions full-length coaching material. The concepts of molecular divergence and molecular clocks form the foundation of modern molecular evolutionary biology, offering insights into the timing and tempo of species divergence and genome evolution. These concepts are rooted in the understanding that mutations accumulate over time in DNA and protein sequences, and that these changes can be used to infer both evolutionary relationships and the timing of divergence between lineages. Molecular divergence refers to the accumulation of genetic differences, in nucleotide or amino acid sequences, between species, populations, or genes, as a result of mutation, genetic drift, and sometimes natural selection. When two lineages diverge from a common ancestor, their DNA sequences begin to differ as mutations accumulate independently. Over time, these differences, measurable as sequence divergence, reflect the evolutionary distance between the lineages. Divergence can be calculated for different molecular markers such as mitochondrial DNA, mtDNA, ribosomal RNA, rRNA, coding and non-coding regions, depending on the resolution required. Molecular divergence is not uniform across the genome. Some regions evolve rapidly, accumulating many substitutions and being useful for studying closely related species, e.g., mitochondrial genes or introns, while other regions evolve slowly, preserving ancient evolutionary relationships, e.g., rRNA genes or histone genes. The divergence is quantified in terms of substitutions per site, and it is often corrected for multiple hits using models of sequence evolution, e.g., Jukes-Cantor, Kimura 2-parameter. The idea of molecular clocks emerges directly from this pattern of divergence. First proposed by Emil Zuckerkandl and Linus Pauling in the 1960s, the molecular clock hypothesis states that mutations accumulate in genomes at an approximately constant rate over time, making it possible to use the number of molecular differences between sequences as a clock to estimate the time since their last common ancestor. This assumption enables the construction of time-calibrated phylogenies and the estimation of divergence times even in the absence of fossil records. A molecular clock is calibrated using known reference points, typically fossil ages or geological events, to convert genetic distances into chronological estimates. For instance, if two species have a genetic distance of 0.02 substitutions per site and are known from the fossil record to have diverged 5 million years ago, the molecular clock rate would be calculated as 0.004 substitutions per site per million years. This calibrated rate can then be applied to estimate divergence times for other lineages. However, real molecular clocks are not perfectly constant, and several factors can lead to rate heterogeneity among lineages. These include differences in generation times, metabolic rates, DNA repair mechanisms, and selective pressures. For instance, species with shorter generation times tend to accumulate mutations faster. To account for such variability, molecular clocks are refined into relaxed molecular clock models, which allow for variable rates across different branches of a phylogenetic tree. In molecular clock analyses, synonymous substitutions which do not change the amino acid sequence are typically preferred as molecular clock markers because they are assumed to evolve neutrally, unaffected by selection. Mitochondrial DNA is also commonly used due to its relatively high mutation rate and maternal inheritance, which simplifies lineage tracing. Molecular divergence and clocks together have profound applications. They allow dating of speciation events, even in the absence of reliable fossils. They help trace human ancestry and migration, as seen in studies of mitochondrial Eve and Y chromosomal atom. They provide temporal frameworks for understanding viral evolution, such as in HIV or influenza tracking. They help in phylogeographic studies, correlating genetic divergence with geographical and climatic changes. In summary, molecular divergence quantifies how much genetic difference has accumulated between sequences, and molecular clocks interpret this divergence in the context of time, transforming sequence data into powerful tools for dating and reconstructing the tree of life. Their utility lies in combining genetic variation with temporal scales, thereby allowing researchers to probe the deep past of biological evolution with unprecedented precision. Previous year CSIR net questions. The molecular clock hypothesis is best supported by which of the following statements? All genes evolve at the same rate. B. Molecular changes accumulate at a constant rate over time. C. Phenotypic changes reflect genotypic changes. The evolutionary rates are determined solely by natural selection. Answer. B. Molecular changes accumulate at a constant rate over time. CSIR net December 2015. Which of the following markers is most suitable for estimating divergence time between closely related species? A. 18sRRNA. B. Histone genes. C. Mitochondrial DNA. D. Ribosomal DNA. Answer. C. Mitochondrial DNA. CSIR net June 2017. You are given a phylogenetic tree based on molecular data. If you want to infer the time of divergence between species, what additional information is required? A. Population size. B. Mutation rate. C. Recombination rate. DGC content. Answer. B. Mutation rate. CSIR net Dec 2019. Let me know when you're ready for the next topic, such as rate of molecular evolution, DNA sequence variation, or phylogenetic tree construction. All will be prepared at maximum depth to serve as your sole study material. You said. Molecular tools and phylogeny. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on molecular tools and phylogeny. Maximum detail. Paragraph format. No tables. CSIR JRF level, based on previous year questions. Full-length coaching material. Phylogenetics, the study of evolutionary relationships among organisms, has been revolutionized by the advent of molecular tools. Traditionally based on morphology and fossil records, phylogenetic inference now relies heavily on molecular data, which offers more objective and quantifiable information, especially for organisms with few morphological differences or incomplete fossil histories. Molecular tools and phylogeny refer to the various genetic markers, sequencing technologies, and computational methods used to reconstruct evolutionary trees, phylogenies based on similarities and differences in genetic sequences. Foundation of molecular phylogenetics lies in the comparison of homologous DNA, RNA, or protein sequences across different organisms. These sequences accumulate mutations over time, and the pattern of shared mutations reflects the evolutionary history of the organisms. Sequences that are highly conserved suggest a recent common ancestor, while greater sequence divergence implies longer evolutionary separation. To extract phylogenetic information, one first needs to select an appropriate molecular marker, amplify it using PCR, and sequence it using high-throughput sequencing or traditional Sanger sequencing. Among the most widely used molecular markers are ribosomal RNA genes, such as 16sRRNA in prokaryotes and 18sRRNA in eukaryotes, which evolve slowly and are useful for deep evolutionary comparisons across phyla and kingdoms. On the other hand, mitochondrial genes like COI, CYTB, and chloroplast genes are used for resolving relationships among more closely related species due to their higher mutation rates. Nuclear genes, including single-copy orthologous genes, are also highly informative in phylogenetic studies, especially for eukaryotes. Furthermore, non-coding sequences, introns, and intergenic spacers may provide insights into recent divergences due to their relatively rapid evolution. Once sequences are obtained, they are aligned to identify homologous nucleotide or amino acid positions, a critical step before tree construction. Misalignment can lead to incorrect phylogenies. Alignment tools like Clustol Omega, MAFFT, or Muscle are typically used. The aligned sequences are then analyzed using various tree-building algorithms, each based on different models of molecular evolution. Three major approaches dominate molecular phylogenetic tree construction: distance-based methods, character-based methods, and statistical methods. In distance-based methods like neighbor joining, NJ, or UPGMA, evolutionary distances between sequences are calculated and used to construct a tree that best fits those distances. These methods are computationally fast and suitable for large datasets but may oversimplify the underlying evolutionary processes. Character-based methods include maximum parsimony, MP, and maximum likelihood, ML. Maximum parsimony searches for the tree that requires the fewest evolutionary changes. It is simple and intuitive but often less accurate when substitution rates vary across lineages. Maximum likelihood, in contrast, uses statistical models of sequence evolution like Jukes Cantor, Kimura 2 parameter, or GTR to find the tree that is most probable given the data in the model. Though computationally intensive, ML is among the most accurate methods and is widely used in current research. Another robust method is Bayesian inference, implemented in tools like Mr. Bayes and Beast, which uses prior probabilities and likelihood framework to estimate the posterior distribution of phylogenetic trees. Bayesian methods provide estimates of uncertainty and are particularly useful in time-calibrated phylogenies, integrating fossil or molecular clock data to infer divergence times. Support for the resulting phylogenetic relationships is assessed using bootstrap analysis, which tests the robustness of tree topology by resampling the data multiple times. Bootstrap values above 70% are generally considered reliable. Other support measures include posterior probabilities in Bayesian analysis and jackknife resampling. Modern tools such as Mega, RAXML, PhyML, IQ-Tree, and Beast have made molecular phylogenetic analysis accessible and standardized. High-throughput sequencing and genome-wide phylogenomics have further elevated the resolution of phylogenetic trees, especially in resolving complex evolutionary questions such as ancient radiations or horizontal gene transfer events. In summary, molecular tools in phylogeny include the selection of appropriate genetic markers rRNA, mtDNA, nuclear genes techniques for amplification and sequencing, software for sequence alignment and tree construction, and robust statistical models for evolutionary inference. These tools together allow for accurate reconstruction of evolutionary history, even when morphological or fossil data are limited, and have transformed fields ranging from systematics to conservation biology. Previous year CSIR net questions. Which of the following genes is most appropriate for constructing a phylogeny of distantly related eukaryotes? A mitochondrial COI gene. B. Nuclear intron sequence of C18srRNA gene D. Cytochrome B gene. Answer: C18srRNA gene, CSIR net June 2016. In molecular phylogenetics, the purpose of multiple sequence alignment is to a design primers for PCR. B. Determine gene expression. C. Identify homologous positions for comparison. D. Estimate GC content. Answer: C. Identify homologous positions for comparison. CSIR net Dec 2018. Which of the following methods is based on statistical modeling and is considered most accurate for phylogenetic reconstruction? A. UPGMA. B. Maximum parsimony. C. Maximum likelihood. D. Neighbor joining. Answer: C. Maximum likelihood. CSIR net June 2017. Let me know when you're ready for the next lecture, such as gene duplication and orthology virology, or genome evolution. All will be crafted at the same JRF cracking quality. You said. Classification and identification, ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on classification and identification. Maximum detail, paragraph format, note tables, CSIR JRF level, based on previous year questions, full-length coaching material. Classification and identification are fundamental components of systematics, the scientific study of organismal diversity and the relationships among organisms through time. These processes are critical not only for cataloging biodiversity but also for organizing knowledge in a way that reflects evolutionary relationships. Classification refers to the process of grouping organisms into hierarchical categories based on shared characteristics, while identification is the process of assigning an organism to an existing taxon based on its traits. The basis of biological classification has evolved from purely morphological and anatomical traits to include molecular, biochemical, embryological, ecological, and behavioral characters. Modern classification systems are phylogenetic, meaning they attempt to reflect evolutionary descent and genetic relatedness. Organisms are grouped into taxa such as domain, kingdom, phylum, class, order, family, genus, and species, with species being the most specific and biologically meaningful unit. These groupings are based on shared derived characters, synapomorphies, which are homologous features inherited from a common ancestor. Identification involves comparing an unknown organism to known reference taxa using diagnostic features. Classical methods include the use of dichotomous keys, field guides, monographs, and flora spawnas, which rely heavily on morphological features. However, morphology alone can sometimes be misleading due to convergent evolution, phenotypic plasticity, and cryptic species organisms that are morphologically indistinguishable but genetically distinct. Hence, identification increasingly incorporates molecular tools, including DNA barcoding, which uses a short genetic sequence, commonly COI gene in animals and MACK or RBCL genes in plants, to uniquely identify species. Molecular phylogenetics plays a critical role in classification, especially in resolving relationships where morphology is inadequate or misleading. Genes used for this purpose include both conserved genes for deeper evolutionary relationships, e.g., 18srRNA, and rapidly evolving genes for resolving recent divergences. The integration of molecular data with traditional taxonomic methods is called integrative taxonomy, which ensures greater accuracy and species delimitation in classification. In taxonomy, nomenclatural codes such as the International Code of Zoological Nomenclature (ICZN) and the International Code of Nomenclature for Algae, Fungi, and Plants (ICN) govern the naming of organisms. Each valid species name must be published with a type specimen, a unique identifier that serves as the reference for the application of that name. Taxonomic classification systems have undergone significant changes, particularly with the advent of molecular data. Traditional five-kingdom systems Monera, Protista, Fungi, Plantae, Animalia have been largely replaced by three-domain systems Archaea, Bacteria, Eukarya, and further subdivided into numerous clades based on molecular phylogenies. This shift emphasizes monophyletic groupings, i.e., groups that include an ancestor and all of its descendants. The concept of species also underpins classification. The biological species concept defines species based on reproductive isolation, while phylogenetic species concept emphasizes genetic distinctiveness and diagnosable traits. In microorganisms, where sexual reproduction is absent or infrequent, species are often delimited using genetic similarity thresholds, e.g., 97% similarity in 16srRNA. Overall, classification provides a stable and universal framework to organize biological diversity, while identification enables the recognition and cataloging of organisms in nature and laboratories. These practices are indispensable in biodiversity conservation, ecological studies, agriculture, and bioprospecting. Previous year CSIR net questions. Which one of the following is the most appropriate method to identify cryptic species? A dichotomous key. B. Morphological comparison. C. Karyotyping. D. DNA barcoding. Answer: D. DNA barcoding. CSIR net June 2016. Which concept of species emphasizes reproductive isolation? A. Morphological species concept. B. Ecological species concept. C. Phylogenetic species concept. D. Biological species concept. Answer: D. Biological species concept. CSIR net Dec 2018. Which of the following is a taxonomic rank higher than genus and lower than order? A. Species. B. Family. C. Phylum. D. Class. Answer: B. Family. CSIR net June 2017. Let me know when you want the next lecture, such as species concepts, systematics and taxonomy, or any topic from molecular biology, cell biology, or ecology. I'll maintain the same in-depth, CSIR JRF cracking quality. You said, protein and nucleotide sequence analysis. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on protein and nucleotide sequence analysis. Maximum detail, paragraph format, note tables, CSIR JRF level, based on previous year questions, full-length coaching material. Protein and nucleotide sequence analysis form the foundation of modern molecular biology and bioinformatics. These analyses are essential to understand gene and protein function, evolutionary relationships, and molecular mechanisms governing life. In the context of the CSIR-NET JRF Life Sciences exam, this topic frequently appears with emphasis on sequence alignment, similarity search tools, scoring matrices, and evolutionary inference. Nucleotide sequence analysis involves the study of DNA and RNA sequences to identify genes, regulatory elements, and motifs, as well as to compare sequences across organisms to infer evolutionary and functional insights. One of the primary steps in nucleotide analysis is sequence alignment, where DNA sequences are arranged to identify regions of similarity. These similarities may indicate functional, structural, or evolutionary relationships between the sequences. In pairwise alignment, two sequences are compared either globally, aligning from end to end using the Needleman-Wunsch algorithm, or locally, aligning subregions using the Smith-Waterman algorithm. Global alignment is suitable for sequences of similar length and content, while local alignment is better for finding conserved domains within divergent sequences. For multiple sequence alignment (MSA), where 3 or more sequences are compared, tools like Clustal Omega, Muscle, and MAFFT are commonly used. MSA helps identify conserved motifs, functional residues, and can aid in phylogenetic analysis. Following alignment, similarity or identity percentages are computed, which provide quantitative measures of how closely related two sequences are. Scoring matrices are used to assign values to matches, mismatches, and gaps in alignments. For nucleotide sequences, a simple match-mismatch scoring system may be used. However, for protein sequence analysis, more sophisticated matrices such as PAM, point-accepted mutation, and BLOSUM, block substitution matrix, are employed. PAM matrices are based on evolutionary models and are suitable for closely related sequences, while BLOSUM matrices, derived from conserved protein blocks, are better for more divergent sequences, e.g., BLOSUM 62 is a standard default in many analyses. Protein sequence analysis extends beyond alignment. It involves predicting secondary and tertiary structures, identifying functional domains, e.g., kinase domains, zinc fingers post-translational modification sites, and subcellular localization signals. Tools such as BLASTP, InterProScan, and FAM allow identification of homologous proteins and functional motifs. Homology-based annotation uses similarity to known proteins to predict the function of newly sequenced proteins. BLAST Basic Local Alignment Search Tool is one of the most widely used tools for sequence comparison. Variants include BLASTN, nucleotide versus nucleotide, BLASTP, protein versus protein, BLASTX, translated nucleotide versus protein, and TBLASTN, protein versus translated nucleotide. The output includes a list of hits with their e-values, expectation values, which indicate the statistical significance of the match. Lower e-values mean more significant alignments. A typical threshold for significance is E and LT, 1E5. Beyond alignment, sequence annotation is the process of adding functional information to raw sequences. In genomic projects, gene prediction algorithms, e.g., GENSCAN, Augustus, are used to identify coding regions, exons, introns, promoters, and other regulatory features. Tools like ORFFinder help locate open reading frames in nucleotide sequences. For proteins, functional annotation can be inferred using databases such as UniProt, SwissProt, FAM, and ProCyte. Sequence analysis also plays a vital role in comparative genomics, where sequences from different species are compared to understand conservation and divergence of genes and regulatory elements. In evolutionary biology, sequence data is used to build phylogenetic trees, estimate divergence times, and detect positive or purifying selection through synonymous and non-synonymous substitution rates (DS and DN). In clinical research and biotechnology, sequence analysis is used in mutation detection, genotyping, SNP analysis, and drug target identification. In ecology and conservation biology, it supports DNA barcoding and environmental DNA and monitoring for species identification and biodiversity assessment. In summary, protein and nucleotide sequence analysis encompasses alignment, similarity search, functional prediction, structural analysis, and evolutionary inference. These analyses enable researchers to decode molecular information encoded in genes and proteins and are indispensable tools in modern biological research and biotechnological applications. Previous year CSIR-NET questions. Which of the following tools is most suitable for identifying conserved domains in a protein sequence? A. BLASTN. B. InterProScan. C. ORFFinder DT Coffee. Answer: B. InterProScan, CSIR-NET Deck 2018. Which scoring matrix is more appropriate for aligning distantly related protein sequences? A. PAM30. B. BLOSUM 80. C. BLOSUM 45. D. PAM1. Answer: CBLOSUM 45, CSIR-NET June 2017. In a BLAST output, a low E-value indicates a poor alignment. B. High sequence similarity. C. Non-homologous sequences. D. Longer alignment length only. Answer: B. High sequence similarity, CSIR-NET June 2015. Let me Which of the following tools is most suitable for identifying conserved domains in a protein sequence? A. BLASTN. B. InterProScan. C. ORFFinder DT Coffee. Answer: B. InterProScan, CSIR-NET Deck 2018. Which scoring matrix is more appropriate for aligning distantly related protein sequences? A. PAM30. B. BLOSUM 80. C. BLOSUM 45. D. PAM1. Answer: CBLOSUM 45, CSIR-NET June 2017. In a BLAST output, a low E-value indicates a poor alignment. B. High sequence similarity. C. Non-homologous sequences. D. Longer alignment length only. Answer: B. High sequence similarity, CSIR-NET June 2015. Let know which topic you want next. I can cover phylogenetic tree construction methods, gene prediction, comparative genomics, or any specific unit from CSIR-NET JRF Life Sciences syllabus in the same high-quality coaching style. You said. Origin of new genes and proteins, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on origin of new genes and proteins. Maximum detail, paragraph format, no tables, CSIR JRF level, based on previous year questions, full-length coaching material. The origin of new gene proteins is a central theme in evolutionary biology, shedding light on how novel functions arise and how genetic diversity contributes to phenotypic complexity. Genes and proteins evolve over time through a range of molecular mechanisms that generate raw genetic material, which is then shaped by natural selection, genetic drift, and other evolutionary forces to produce new functions. These innovations are critical for the emergence of new traits, adaptation to environments, and the diversification of life forms. One of the most well-established mechanisms for the origin of new genes is gene duplication. When a gene is duplicated, the two copies paralogs may initially have redundant functions. However, over evolutionary time, one copy may accumulate mutations that allow it to diverge in function. This can lead to neofunctionalization, where one copy acquires a novel function, or subfunctionalization, where both copies partition the original function. Tandem duplications, segmental duplications, and whole genome duplications, e.g., in plants and early vertebrate evolution, are major sources of gene duplication. These duplicated genes are raw material for evolutionary innovation. Exon shuffling is another process by which new genes and protein domains arise. In this mechanism, exons coding for functional protein domains are recombined in new arrangements through non-homologous recombination, intronic recombination, or retrotransposition. This can result in chimeric proteins with novel domain architectures and functions. The origin of new genes and proteins is a central theme in evolutionary biology, shedding light on how novel functions arise and how genetic diversity contributes to phenotypic complexity. Genes and proteins evolve over time through a range of molecular mechanisms that generate raw genetic material, which is then shaped by natural selection, genetic drift, and other evolutionary forces to produce new functions. These innovations are critical for the emergence of new traits, adaptation to environments, and the diversification of life forms. One of the most well-established mechanisms for the origin of new genes is gene duplication. When a gene is duplicated, the two copies paralogs may initially have redundant functions. However, over evolutionary time, one copy may accumulate mutations that allow it to diverge in function. This can lead to neofunctionalization, where one copy acquires a novel function, or subfunctionalization, where both copies partition the original function. Tandem duplications, segmental duplications, and whole genome duplications, e.g., in plants and early vertebrate evolution, are major sources of gene duplication. These duplicated genes are raw material for evolutionary innovation. Exon shuffling is another process by which new genes and protein domains arise. In this mechanism, exons coding for functional protein domains are recombined in new arrangements through non-homologous recombination, intronic recombination, or retrotransposition. This can result in chimeric proteins with novel domain architectures and functions. Many multi-domain proteins, such as transcription factors and kinases, are believed to have arisen through exon shuffling. Retroposition, or retroduplication, involves the reverse transcription of an mRNA transcript and its insertion into a new location in the genome. These retrocopies are often intronless and may initially be non-functional pseudogenes, but under certain circumstances, they may acquire regulatory elements and evolve into functional genes, retrogenes. Horizontal gene transfer, HGT, although rare in multicellular eukaryotes, is a significant source of new genes in prokaryotes and unicellular eukaryotes. Through mechanisms such as transformation, transduction, and conjugation, organisms can acquire genes from unrelated species, leading to rapid acquisition of new traits such as antibiotic resistance or metabolic capabilities. De novo gene birth is a more recently appreciated phenomenon where entirely new protein-coding genes evolve from previously non-coding DNA sequences. Initially, these sequences may be transcribed and translated as short open reading frames, ORFs, which might then acquire beneficial mutations. If the peptides they encode confer a selective advantage, these sequences may be retained and refined into functional genes. De novo gene evolution highlights the plasticity of genomes and their capacity to innovate without requiring existing templates. Transposable elements, TEs, also play a critical role in the evolution of new genes. They can disrupt gene regulation, provide new promoters or enhancers, or mediate genomic rearrangements that bring distant sequences together. Some genes are believed to have originated from transposon sequences themselves, e.g., the RAG1 and RAG2 genes involved in VT-J recombination in the vertebrate immune system. At the protein level, the evolution of new proteins often involves point mutations, domain duplications, fusion events, and structural rearrangements. Protein structure is more conserved than sequence, allowing for functional innovations without drastic changes to folding patterns. Functional divergence after duplication may be driven by positive selection, especially at sites critical for binding or catalysis. Molecular evolution tools such as deci-Newton per deci-Siemens ratios, non-synonymous to synonymous substitution rates, help detect selection acting on new genes. A deci-Newton per deci-Siemens in GT1 suggests positive selection, often indicative of functional innovation in new genes or proteins. In essence, the origin of new genes and proteins is not a singular event but a cumulative process driven by genomic flexibility, mutation, duplication, recombination, and selection. These events collectively contribute to the functional and regulatory complexity of organisms and play a fundamental role in evolution and speciation. Previous year CSIR net questions. A new gene that originates entirely from non-coding DNA is termed as a pseudogene. B. Retrogene C. de novo gene D. paralog. Answer: C. de novo gene, CSIR net June 2021. The most common mechanism of new gene formation in eukaryotes is a mutation in non-coding RNA. B. Gene duplication followed by divergence C. Horizontal gene transfer D. Frame shift mutations. Answer: B. Gene duplication followed by divergence CSIR net DEC 2017. Which of the following mechanisms does not directly contribute to the formation of novel proteins? A. Exon shuffling. B. Domain fusion C. Synonymous substitution D. Gene duplication. Answer: C. Synonymous substitution CSIR net June 2018. Let me know which topic you'd like next, for example, phylogenetic tree construction, comparative genomics, gene regulation in prokaryotes, or any other CSIR unit, and I'll maintain this maximum quality format. You said, gene duplication and divergence. ChatGPT said. Where is your CSIR net JRF Life Sciences lecture on? Gene duplication and divergence. Ultra detailed paragraph format, no tables, CSIR JRF level, based on PYQs, maximum word limit, this is your only coaching material. Gene duplication and divergence is one of the most powerful evolutionary mechanisms for generating novel genes and functions. Unlike point mutations that modify existing sequences, duplication creates an entirely new copy of a gene, which can then evolve independently. This process has played a fundamental role in the evolution of complex genomes, especially in eukaryotes, and underlies many innovations in metabolic pathways, development, immune systems, and sensory perception. Gene duplication occurs when a segment of DNA containing a gene is copied and inserted into another location in the genome. This may happen through unequal crossing over during meiosis, replication slippage, retrotransposition, segmental duplication, or even whole genome duplication (WGD). In unequal crossing over, homologous chromosomes misalign during recombination, leading to one chromosome with an extra gene copy and the other with a deletion. In retrotransposition, an mRNA transcript is reverse transcribed and inserted back into the genome. These copies typically lack introns and regulatory elements. Segmental duplications can duplicate not just single genes but entire clusters or genomic regions, while whole genome duplication events, especially common in plants and early vertebrates, double the entire gene content, providing massive potential for innovation. After duplication, gene divergence occurs through the accumulation of mutations in one or both gene copies. Since the original function is already preserved by the original gene, the duplicate is free from purifying selection and can tolerate neutral or even deleterious mutations. This relaxed selection is the basis for innovation. Several evolutionary fates are possible for duplicated genes. Non-functionalization, pseudogenization. The duplicate gene acquires disabling mutations and becomes a non-functional pseudogene. This is the most common fate of duplicated genes. Subfunctionalization. The ancestral function is split between the two duplicates, with each retaining a subset of the original function. This is often explained by the duplication degeneration complementation (DDC) model, where regulatory or functional constraints are partitioned. Neofunctionalization. One copy retains the original function, and the other acquires beneficial mutations that confer a new function. This is a rare but important pathway for the evolution of novel traits. The divergence of gene duplicates can also be measured using Deci-Newton per Deci-Siemens ratios, non-synonymous to synonymous substitutions. A ratio in GT, 1 suggests positive selection, adaptive evolution, and LT, 1 implies purifying selection, and approximately 1 indicates neutral evolution. Gene families, such as olfactory receptors, cytochromes P450, globins, and immunoglobulins, have evolved through extensive duplication followed by divergence. For example, the globin gene family, including hemoglobin and myoglobin, originated via duplication events followed by divergence in both sequence and expression pattern. Some genes became expressed in the embryonic stage, while others in adults. An important consequence of gene duplication is functional redundancy, where both genes perform the same role. Over time, this can be resolved by divergence or by retaining both copies for dosage balance, especially in protein complexes or stoichiometrically balanced pathways. Dosage-sensitive genes tend to be retained in duplicates, as seen in ribosomal proteins and transcription factors. Duplicates can also evolve tissue-specific expression, allowing finer regulatory control and specialization. From a genomic perspective, gene duplication contributes significantly to genetic robustness, evolvability, and biological complexity. New regulatory elements can evolve in duplicated genes, leading to altered expression patterns. This is evident in developmental genes like Hox clusters, which expanded via duplication and underlie the diversification of body plans and metazoans. Modern tools in comparative genomics and transcriptomics help trace gene duplication and divergence events across lineages. Whole genome sequencing has revealed that ancient duplication events, including 2 rounds of whole genome duplication, 2R hypothesis in early vertebrates, played key roles in the evolution of vertebrate complexity. In conclusion, gene duplication and divergence is a cornerstone of molecular evolution. It provides raw material for evolution to experiment with new functions while retaining the original gene function. The balance of mutation, selection, and genomic architecture determines the evolutionary fate of gene duplicates and the emergence of new traits. Previous year CSIR-NET questions. Which of the following statements best describes subfunctionalization following gene duplication? A. One gene becomes non-functional. B. Both genes acquire new functions. C. Both genes lose part of the original function but together retain the full ancestral function. D. The duplicate gene is deleted. Answer. C. Both genes lose part of the original function but together retain the full ancestral function. CSIR-NET Dec 2016. The globin gene family in vertebrates is an example of a lateral gene transfer. B. Neofunctionalization. C. X and shuffling. D. Subfunctionalization only. Answer. B. Neofunctionalization. CSIR-NET June 2018. Which evolutionary mechanism is most likely responsible for the expansion of gene families such as cytochrome P450 and olfactory receptors? A. Point mutations. B. Horizontal gene transfer. C. Gene duplication. D. X and skipping. Answer. C. Gene duplication. CSIR-NET June 2020. Let me know which topic you'd like next. I recommend phylogenetic tree construction, comparative genomics, or we can move to gene regulation in prokaryotes or transcriptional control in eukaryotes, depending on your CSIR-NET preparation plan.